Okay. So the first thing that you have to know about the eye is that now the entire of this structure here is this is the eyeball. You can see it is oval. And what you see is just a small portion of the entire eyeball. That's what is in front. Another thing that I want you to understand before you can even go much further is that you are going to see two chambers or two spaces or cavities on the eye. So this white thing that you can see here, this is the lens. The lens is attached using these fibers. We call them zonular fibers to the ciliary body. And the cavity that is posterior to the lens, we call it the posterior cavity. The cavity in front of the lens, we call it the anterior cavity. The posterior cavity has got a fluid which we call the vitreous humor. It is jelly-like. The anterior cavity has got a fluid which we call the aqueous humor. It is aqueous even from the word. It's like water. You'll be able to see the fluid in head and neck next year. So again, when you look at the anterior cavity, you have got a lens. This sorry, not the lens, but the iris. Is there a better picture here? Okay, hope you can just see this one. So this is the iris. It's supposed to be going like that. The iris is in front of the lens, and the iris is going to leave a space on the middle. That is known as a pupil. It's just a space. So the space between the iris, the front of the eye, this is the cornea. The space between the iris and the cornea, that's known as the anterior chamber. So the anterior cavity has got the anterior chamber and the posterior chamber. Remember, I've got the posterior cavity behind the lens, anterior cavity in front of the lens. But just in front of the lens, you are going to have two cavities which are being separated by the iris. In front of the iris, you have got the anterior chamber. Behind the iris, you've got the posterior chamber. Importance of that is that behind the posterior chamber on the ciliary body here, it is the ciliary body that will produce aqueous humor. The aqueous humor then is going to flow, and then it is going to pass through the pupil, and then it will drain in front of the iris. You are going to have a canal here which is known as a canal of Schlemm. That is where the aqueous humor will be able to drain. So when you are talking about the eye bulb, it has got three layers. You have got the fibrous layer, which is the outermost layer that you can see here. You have got the vascular layer and the neural layer. The fibrous layer is also known as avascular because it doesn't have any blood vessels. Okay, the outermost layer. And what you need to know about the outermost layer is that it has got two layers. The anterior 1/6, 1/6. In front, you have got the cornea. This is the part that is in front, the cornea, and it is 1/6. While the posterior aspect is going to be the sclera. That is basically the whitest thing that you'll be able to see. Of course, the cornea is the one that is covering the iris and the pupil, because the pupil is actually a space. So without the cornea, you could have used a needle to take it inside to reach to where the lens is. But because of the cornea, it is transparent. You can see through it. That's why you can see a kama pupil on the middle of your eye. When you come, so the this structure, the fibrous tunic, as we have said, it doesn't have any blood vessels. So therefore, meaning that for it to remain nourished and with oxygen, it's going to be able to receive those nutrients by diffusion from the layer that has got the oxygen and the nutrient. If you come to the vascular layer, we call it vascular because it is now having blood vessels. So you are going to have the choroid and the ciliary body. These are continuations of each other. So this is more inside. Remember, the outermost was the fibrous layer, which had the behind sclera, front cornea, and then as you get to go inside, you're going to have the vascular layer. The vascular layer is going to have this, which is known as the choroid. Okay, you can even see it here, the choroid. And then as you go in front, the choroid is going to give you the ciliary body and also the iris. So take note of that. And then now you're going to have the neural layer, which is the innermost layer. And this layer is basically made up of the retina. Now, the retina has got cells, we call them photoreceptors, that will be able to pick up sensations. If I touch you, you can feel my touch because you have got mechanoreceptors that will be able to pick up those sensations. Same with the tongue. When you taste anything, you'll be able to pick up those sensations because there are receptors on the tongue. So a stimulus always binds to a receptor, and then that will generate an action potential that is going to communicate to the brain to be able to tell what kind of sensation that is. So the neural layer, which is the retina, has got the rods and the cones. And those guys are vital because they are the ones that are responding to light. Do you have any question up to this end? Okay. So let's start with the cornea then. The cornea, you can see it is the front aspect of the fibrous layer. And the thing is, this will be way, way, way easier if you understand from here. You know why? Because next year in neuroscience, these are going to be topics you'll be doing in neurophysiology, in neuroanatomy. So meaning if you understand here, it will be way, way, way easy. So the cornea is a transparent, avascular, and highly innervated anterior portion of the fibrous cord. You can see that it is avascular, but it has got innervation, meaning that there are some nerves that will be able to control it there. Okay. So the epithelium of the cornea is the same as the epithelium of the skin, except the cornea is not dry, all right, it's wet. So it is non-keratinized, stratified squamous epithelium, the same epithelium found in the mouth, the esophagus, the vagina, and it contains numerous free nerve endings. Because it contains free nerve endings, these are receptors for pain, meaning that the cornea can feel pain. Remember, when it comes to viscera, internal organs, they don't feel pain, but cornea can feel pain. Okay. You also have what we call the Bowman's membrane. This is histologically. It is a homogeneous non-cellular layer containing type 1 collagen fibers. If you can see on top, you have got the epithelium on this slide, and then down you are going to have the Bowman's membrane, which is a homogeneous non-cellular, doesn't have any cells, which contains type 1 collagen fibrils. And then you have got the stroma, which is a supporting tissue, and it is the thickest layer, 90%. It has got parallel lamina of dense collagenous connective tissue, which have been regularly arranged to make it transparent. And each lamina, each one of these lines for these connective tissues is composed of mainly type 1 collagen fibers with long fibroblasts. And then you are going to have the descent membrane or Descemet's membrane, which is a thick basement membrane where these cells on top are going to be able to rest. You call that as a Descemet's membrane. And then now we're going to have the corneal endothelium. Endothelium is basically epithelium that lines inner cavities like of blood vessels. Also the heart, except we will call it mesothelium. So it is a simple squamous epithelium If you just hear of endothelium, that's simple squamous epithelium. The function is it helps in dehydration. Okay, so dehydration will help in transparency. That's why the eye is going to be open like that. So the corneal epithelium is involved in formation of food basement membrane, which is the thick basement membrane on top, and then keeping the stroma, which is on top, relatively dehydrated without any water. So there is a sodium pump that will cause water withdrawal from the stroma because of this. The corneal endothelium, remember we are saying that it keeps the stroma relatively dehydrated. So because of these sodium pumps, and when you are removing sodium from the stroma, where sodium goes, that is where water also goes. So you are going to be pulling the water away, and that dehydration is what makes this cornea to be transparent. You can see through it. Okay. So we have got what we call the corneoscleral junction. Even from the word, it's basically the junction between the cornea and the sclera. So the sclera is the posterior aspect, the cornea is the anterior 1/6, the sclera is posterior 5/6. So it's also known as the limbus. It's a transition region between the cornea and the sclera, and it's about 1.5 millimeters in width. And this place is highly vascular. It has got blood vessels, and that to provide nutrition to the cornea, which is avascular, right? So what is found on the limbus or the corneoscleral junction? We have got the trabecular meshwork, which is endothelium lined spaces. And these spaces, remember what I mentioned, the canal of Schlemm, that is where the aqueous humor will be able to drain. It is a meshwork. Think of a sheave, right? In those olden days, you would be you would pound some mini meal, and then you get a sheave so that you remove bigger particles. So that is how this place is. It is a trabecular meshwork where the aqueous humor will be draining. In case the aqueous humor has got some dirt, that date can be removed. So you need to know a clinical colorate just on the canal of Schlemm, which drains the aqueous humor. Sometimes, can you see the angle between the cornea and the iris? At least there's an angle there that leaves the canal of Schlemm open. So sometimes the iris will be so close to the cornea such that this angle is closed, and so there will be failure to drain the aqueous humor. Or you can have the canal of Schlemm itself, the meshwork being blocked by some debris, some dirt there, and that will still lead to failure to drain aqueous humor. But then the problem is that you have not stopped producing it. So aqueous humor will start accumulating inside the eye. The normal pressure, we call it intraocular pressure, because the pressure in the eye created by these fluids is 10 to 20 millimeters of mercury. So when there is aqueous humor failure to drain, there's going to be accumulation of this aqueous humor, and that leads to what we call glaucoma. So glaucoma can either be primary or secondary. Secondary is because of a problem somewhere else. Primary is a problem in the eye itself where draining is affected. Now, this glaucoma can either be open angle glaucoma or closed angle. Open angle, the problem is with the meshwork, it is blocked, but the angle here is just okay. Closed angle means the angle here is closed, so you can't drain the aqueous humor, right? Is there any question there? Is there any question? Are we moving together? Someone just to confirm if we are together. Okay, so we go now to the sclera, which is the posterior part of the fibrous layer. The sclera will cover the posterior 5/6, as I already mentioned area of the fibrous tunic or the fibrous layer. So I've got the sclera proper, which has got interlacing bundles of type 1 collagen, which is dense collagenous connective tissue, the irregular type. Okay. And then you also have melanocytes. These melanocytes, remember melanin is important in trapping what? Light. So these melanocytes will be able to help in the trapping of light. Okay. So you can see also the histology here. The sclera is there. You have got the choroid, the retina. Remember, the sclera is the outer layer. It is outer layer. So this space that you can see here is a posterior chamber. So when you're talking about these layers, from the inside going outward, you're going to have retina, choroid, and then sclera. And you can see also the canal of Schlemm at this point where I've got the ciliary body. That is histologically. Now, when you go to the vascular tunic or layer, the choroid, ciliary body, and also the iris, as we mentioned. Choroid is vascular because it's a vascular layer, and at the back it is pigmented. Okay, it is is pigmented posterior portion of the middle vascular tunic. Yes, it's in the anterior chamber. So aqueous humor will be produced in the posterior chamber, and then it will drain into the anterior chamber. Remember, both the anterior chamber and the posterior chamber are in the anterior cavity. Okay, because the posterior cavity has got vitreous humor. Is that okay? Okay. So you can also see it here. This is the ciliary body in the posterior chamber. When it produces aqueous humor will move like this, and then it will pass. There will be a space here where you have got the eye, the pupil, and then it will come like this and then drain in the canal of Schlemm. So the choroid is separated from the retina by Brach's membrane, that is histologically from the retina. Remember, after you have got the fibrous layer, after the fibrous layer, you have got the vascular layer where you find the choroid. After the vascular layer, you are going to have now the neural layer where you're going to find the retina. So between the retina and the choroid, you have got Brach's membrane. And then the ciliary body is the anterior continuation of the choroid, and it surrounds the lens. It's formed of loose vascular and pigmented connective tissue that contains three bundles of smooth muscles, okay, which are ciliary muscles. So this ciliary body has got smooth muscles in it. So the importance, you should know at least the importance. Its inner surface is lined by pars ciliaris retinae, two rows of columnar cells, outer pigmented and inner non-pigmented layer. Its inner surface is highly folded, forming the ciliary process. Basically, what we are saying is this. The ciliary body contains three bundles of smooth muscles, which we call ciliary muscles. It is actually from this, you can see the ciliary muscle like that. if you look at this structure, it's actually from the ciliary body that you're having the zonular ligaments going to attach to the lens. So what happens that the muscles in the ciliary body, if they contract, they are going to cause the zonular ligaments to relax. When the zonular ligaments relax, that will cause the lens to become shorter and thicker on the middle. And the opposite happens when when the ciliary muscles relax, the ligaments will contract, the zonular ligaments. That will pull on the size of the lens. It will make the lens to become longer and thinner. And all that is important in accommodation so that you can be able to see clearly objects which are near and objects that are far. And then the ciliary process will project from the inner surface of the anterior one third of the ciliary body towards the lens and are covered by pars ciliaris retinae, which are two rows of column cells, same like the ciliary body. And they'll give attachment to the suspensory ligaments, which are also called zonular fibers. Okay, it's on the ciliary processes, these things that are coming out like that. When we go to the iris, it's also part of the vascular tunic or vascular layer. Anterior borderline, incomplete layer of fibrous. So the iris has got 5 layers. You have got the first being the anterior borderline, the stroma, the vessel layer, dilator papillae or pupillae, muscle layer, and posterior surface. So the anterior border layer is an incomplete layer of fibroblast and melanocytes. So it has got fibroblasts and melanocytes. The stroma is connective tissue supporting connective tissue, which is poorly vascularized with fibroblasts and melanocytes. Pigments are responsible for eye color. So these melanocytes which are found in the iris would be determining the eye color. The vessel layer is well vascularized because vessel and it has loose connective tissue. Centrally, it contains these circularly arranged smooth muscle fibers. Okay. The dilator papillae should be okay. Let me see if I can try to draw this so that you just make sense out of it. Okay, so let me try to draw so that you get what we are trying to say about the iris here, the muscles that it has. So the iris, when you are looking from the front of the eye, it's going to be like this. It's going to leave a space on the middle, which we call the pupil. It has got some muscles which are round, like that. Those are circular muscles. It also has got muscles which go like that. We call these as radial muscles. If you are looking on the side, what you're going to be able to see is the lens is going to be somewhere here, and then it's attached to the zonular fibers. In front, that's where you're going to have this iris like this. You can see still a space is leaving there. So I've got radial muscles and you have got circular muscles. So what happens is that when the radial muscles contract, these ones which are going straight, they are going to be making the pupil to become big. So radial muscles are also known as the dilator pupillae because they dilate the pupil. Circular muscles they are in circle. If they contract, they will be making the pupil to become small. So they are also known as sphincter pupillae. So the sympathetic nervous system is the fight or flight. When you see a lion or you see your girlfriend at a distance hugging another guy, what will happen is that the sympathetic nervous system is activated. So your eye is going to prepare to be able to see clearly by directing the pupil. So the sympathetic nervous system causes dilatation of the pupil, okay, by affecting the by causing the contraction of the radial muscles and relaxation of the circular muscles. So these guys are antagonists. If one contracts, another one relaxes, just like that. You also have the posterior surface layer, which is composed of two rows of pigmented epithelial cells, the pars iridis retinae, okay, which are basically epithelial cells, columnar in nature, two of them. So these are the layers of the iris: anterior border layer, and then you have got the stroma supporting tissue, the vessel layer, dilator pupillae muscle layer, and then you have got the posterior surface layer. Is there any question? Are we okay? Okay, great. Now, when we go to, so we are done with the fibrous layer, the vascular layer. Now we go to the neural layer, which is basically the retina. The retina has got too many important types of cells. It has got rods and cones. And we call these cells as photoreceptors. Rods are active in dark or dim light. The cones in light. So what happens is this. When you are in the light, the cones are working. The rods have been deactivated. When you move from light, you go to darkness. It will take a bit of time for the rods to be activated. That's why when you move from where it is light to where it is dark, first you'll not be able to see. But the more you spend time there, the more you adapt, you start seeing. Why? Because now the rods have been activated, which allows you to see in the dark. So hope we have understood that. And then again of importance, what you should be able to know is that you have got three important cells. Okay, three important cells when it comes to the retina. They are not the only ones, but they are at least the most important for now. So you have got the photoreceptors. Can you see these cells in blue here? These are the photoreceptors. They are the cones and the rods. These are, so if this is your eyeball, this one. So the cones and rods are going to be the very last cells in the retina. Remember, you also have the sclera, you also have the choroid layers, and then now you came to this layer. Okay, we are going to have the rods and the cones. After the rods and cones, we're going to have other cells, which are these cells which are here. And you call those cells as bipolar cells. And then after those bipolar cells, you are now going to have these cells here. These are the cells that are now going to come together and form the nerve that controls sight or seeing. So those ones we call them ganglion cells. So there is some interesting physiology that we can talk about here, but this is not neurophysiology, so keep it there. But then when light is going to move all the way from here, because it has come from the pupil that side, it will go and hit the end. It will come and hit the end, stimulating these cells. When these cells are stimulated, they are going to release neurotransmitters which will stimulate these other cells, the bipolar cells. When the bipolar cells are stimulated, they also release neurotransmitter that is going to stimulate the ganglion cells. Ganglion cells are now going to carry an action potential that is going to go via the nerve that these cells will be forming. So these will form cranial nerve number 2, which is the optic nerve. Now you can see you also have some other different types of cells amongst these. I remember in last year's practical, was that practical test 2, or if not the exam, they brought this diagram to name all the layers of the retina. So the first thing is that each cell has got a cell body, isn't it? So firstly, before you can have the rods and the cone, you have got, because when it comes to the last layer, remember we've said that you have got 3 layers of the eyeball. You have got the avascular layer, you have got the vascular layer, the avascular also known called the fibrous, and then you have got the inner layer. The inner layer has got 2 layers. It has got the neural layer, it also has a pigmented layer. The pigmented layer is the outermost, this layer that you can see here, that is just next to the choroid. And then after that, now we're going to have the neural layer, which contains the rods and the cones. So starting from the very bottom, you have got the pigment epithelium, that's the first layer of the retina. After the pigment epithelium, now we're going to see the rods and cones. So we're going to have the photoreceptor layer. This is a photoreceptor layer because this is where light is going to come and strike when it comes. After that, you are now going to have outer limiting membrane. So I've got a limiting membrane here. Basically, a distinction where the cell bodies are forming. After this outer limiting membrane, remember this is the outer aspect. This is the inside of the eye. So after the outer limiting membrane, then you are going to have now the cell bodies. Can you see these are the cell bodies? So we're going to have the outer nuclear membrane. So after these cell bodies, you have got this part. Can you see this part is where the bipolar cells and the rods and cones are meeting? So we call that as a plex form. Plexus, they're forming a plexus. So that's the outer plex form layer. Outer plex form layer. After the outer plex form layer, can you see you still have cell bodies here for the bipolars, but it is inside because you also have cell bodies there which are outside. So this is the inner nuclear layer. And then you have having another plexus between the rods, between the bipolar cells and the ganglion cells. So that is an inner plex form layer because we had an outer plex form layer. After that inner plex form layer, you are now going to have, can you see we also have cell bodies of the ganglion cells. So I've got ganglion cell layer. Okay, the ganglion cell layer. And then after the ganglion cell layer, then you are going to have now the nerve fiber layer. Nerve fiber layer, because these are nerve fibers. And then after that, we remember we had the outer limiting membrane. We are now going to have the last layer being the inner limiting membrane. That's where this layer is ending. So these are the 10 layers of the retina. In case they bring a slide or something like that. Now, can you see that you also have some other cells that are used for support? So supporting the ganglion cells and the bipolar cells, you have got what we call Mueller cells. You have got some Mueller cells. These Mueller cells are extending all the way. These are very long cells. Can you see where they are extending from? They're extending all the way from the inner limiting membrane to the very end here. And then you also have the amacrine cells between the rods and the cones and the bipolar cells. So I've already mentioned everything here. This is just a simplified diagram. You can see amacrine cells. The, so I've got cells that can connect the ganglion cells. Those are the amacrine cells. And they are found between the ganglion cells and the bipolar cells. Between the bipolar and the cones or rods, you have got what we call horizontal cells. So take note of that. The Mueller cells are just supportive cells, but connecting these guys, bipolars and the ganglion cells, you have got amacrine cells. And then those which will be able to connect down the photoreceptors to the bipolars will be the horizontal cells. Yes, there's a hand. Can you repeat? Mhm. Okay, so this one is showing the entire retina. This one is showing the entire retina. From here, you can split all the 10 layers are going to be found here, except down here is supposed to have the outer limiting membrane, on top is supposed to have the inner limiting membrane. But remember, this one is just showing us a simplified version of the many cells that you are seeing. You are seeing the rods and the cones, after that, you are seeing the bipolar cells, after that, you are seeing the ganglion cells, and then they'll form the nerve, which is cranial nerve number 2 here. Does that make sense? At the back of the retina, you are going to have an area which is known as the fovea centralis. Fovea centralis lies in the center of what we call macula lutea. Lutea just means yellow. So we're going to have a yellow layer at the back there. And then the center of that macula lutea is what we call the fovea centralis. And this part is what contains the highest number of cones. Okay, the highest number of cones are concentrated on the fovea centralis, and it is responsible for visual acuity. Visual acuity is how far you can see, like how sharp you can see and how far you can see in the sides. Okay, that's your acuity. You can try to put your finger in front of your eye and then start moving it to the side. When you stop seeing it, it is now out of sight. So that's beyond your visual acuity. So what are the types of cells that are found in the retina? Pigmented epithelium, nerve cells, photoreceptors, bipolars, ganglion cells, and also associated neurons like horizontal cells, amacrine cells, the neuroglial cells, the Mueller cells, the astrocytes. A clinical colorate before we even talk about it, you have got the conjunctiva. What is a conjunctiva? It is a transparent mucous membrane lining the inner surface of the eyelid. The inner surface of the eyelid. Remember, the eyelid is down and up. So that mucous membrane that will be able to line them. Okay. Eyelid is also known as the palpebral conjunctiva. So what is epithelium of the conjunctiva? It is stratified columnar epithelium with numerous goblet cells which secrete mucin on the surface of the eye. It also has a lamina propria and connective tissue. So sometimes the conjunctiva can be inflamed. That's inflammation of the conjunctiva. That's known as conjunctivitis. So already talked about. So you are going to have the optic disc when it comes to the eye. The optic disc is just the region, this region where the optic nerve is entering the the retina, and it has got blood vessels coming out from there. That's a most vascularized place of the retina. Adjacent to it will be the fovea centralis found on the macula lutea. The iris, these are pliable structures containing skin, muscles, and conjunctiva that protect the eye. You know the iris, right? So basically, skin and muscle and conjunctiva. Anteriorly, skin with a few small hair and sweaty glands. You have got what we call tarsal plate or tarsus, sebaceous gland, and modified apocrine sweat glands. So the eyelid is going to have these sebaceous glands and modified apocrine sweat glands. The tarsal glands, or which we call meibomian glands, are modified sebaceous glands. And then you have got modified. Remember, sebaceous glands produce sebum that keep the skin uh, looking this fresh without cracking. Produces oil. So I've got the tarsal glands, which are modified sebaceous glands, which we call meibomian glands. And then modified sweat glands, we call them the glands of Moore. They are present in the lid near the free edges. Sebaceous glands in the eyelashes are called the glands of Zeiss. Accessory lacrimal glands present above the tarsal plate are known as glands of Wolfram. These are lacrimal. The accessory produce tear. Lacrimal glands produce tear. Accessory lacrimal glands present above the tarsal plate. These are the glands of Wolfram. So take note of the glands of Moore, which are modified sweat glands, the glands of Zeiss, which are sebaceous glands in the eyelashes, and then the meibomian glands, which are modified sebaceous glands, so called the tarsal glands. So sometimes you can block sebaceous glands and you're going to have sty, okay? Uh, Something that is projecting out like that, like a pimple on the eye. Or chalazion, which is the inflammation of meibomian glands because of blockage. So these glands, they are producing sebum, but that sebum can't be drained because there's a blockage. So you end up with that. Hope you've seen some people. Yes? Okay. So the sebaceous glands, these are just the same modified sebaceous glands in the eye. The glands, the tarsal glands. So on top here, you are going to have a muscle which we call the tarsus muscle, and on top and down the small muscles, but they are inside, they are hidden within these eyelids. So those glands are near there. So we are calling them tarsal glands of Meibomian glands, which are the sweat glands. Even the same sebaceous glands of this, they still can produce sebum. So even them, if they are blocked, to still be the same thing, except for the Meibonian glands, which are modified sebaceous glands. Their blockade is specifically leading to that inflammation that is causing the chalazion. But for these others, which are the glands of this, if these are now, they are not modified, these are just sebaceous glands. So if they get to be blocked, that will cause a sty. Okay, you can see these muscles that we are talking about. You have got this muscle here. It's found in the upper eyelid. We call it, because it elevates the upper eyelid, we call it the levator palpebro-superialis. Levates, see the upper eyelid, that's the palpebro. It's on top superialis. I've got the lacrimal gland on this side, and then you have got this side, the lacrimal sac. This is where tears are going to be drained. The lacrimal gland is going to produce tears. The tears will be able to drain on this side, and this is going to go and connect to what we call the nasolacrimal duct. It is found in the nose. That is why you find that when someone is crying, they are going to have a running nose, because the tears actually are drained through the nose, okay, via the lacrimal apparatus, which has got the puncta, which is the opening, and then the lacrimal canaliculi, which will drain into the lacrimal sac, which goes to the nasolacrimal duct in the nose. Short-sightedness will just mean difficulty in seeing distant object. You are short-sighted, you can only see things which are near. And the problem is also known as myopia. The problem is that the image forms just in front of the eye. So for accommodation to happen, you should be able to bring in a lens that will scatter, at least will divert the light, so that merging or accommodation can happen on the retina. And what you can use there is a concave lens in short-sightedness. It's opposite in long-sightedness. You use a convex lens because the image is forming beyond the retina. So you use a convex lens so that the image can form real quickly. So the parts of the eye that are involved in reflection of light includes the cornea and also the lens. Actually, also the fluid found inside the eye. Okay. You also have what we call astigmatism, which is basically a refractory error in which you have got irregular shaped cornea or lens. So because the lens or the cornea is irregularly shaped, you can see the images are not hitting one point of the retina, instead they are striking different parts. So you can use a cylindrical lens to correct that mistake. Sometimes some people are color blind, they can't differentiate colors, so you can use what we call the Ishihara test. You ask them to read these numbers. If they can read it or they can't read, you can be able to determine. If they can't read, someone is color blind. If they can read, then they are okay. There is more that we can talk about the eye, but then this is not neuroscience. This is basic anatomy. So we'll end here for now. These are some questions that you can just try out. Thank you so much. We'll meet in the evening for another class.